Hi friends, today we are going to see the past year NEET questions for the chapter Photosynthesis, and this video will cover part 4 for you. Now let's begin with the first question here, which was asked in the year 1992. Photosystem 2 occurs in dash. Easy question, right? So what is the photosystem 2 involved in? We know it is comprised of two systems. One is photosystem 1 and photosystem 2. Now how can you describe photosystem 2? See, photosystem 2 also talks about the synthesis of ATP, and they also talk about the they are involved in the photosynthesis. We have chlorophyll, everything involved in it. Now coming to the photosystem 2, see what is the difference between 1 and 2? Photosystem 2 is a specialized protein complex. Okay, that is the difference between both the systems. You always have to remember it is a protein complex that uses light energy to drive the transfer of electrons from water to the plastoquinone, resulting in the production of oxygen and the release of reduced plastoquinone into the photosynthetic membrane. So what happens in photosystem 2? That is very important. See, it is the first link in the chain of photosynthesis. As I already said, it is involved in the process of photosynthesis. This is the first link in the chain of photosynthesis. Okay, in the chain of photosynthesis. Other than that, it captures photons. Okay, photons are what? They are light. Captures photons and uses the energy to extract electrons from water molecules. First, when the electrons are removed, the water molecules are broken down into oxygen gas, which bubbles away, and hydrogen ions, which are used as to power the ATP synthesis. Now this process, where does it take place? If you see, it takes place, or it is located in the part of the grana-thylakoids. Okay, so correct answer is option C. So here you have the grana, which I show here. You can see it's like disc shaped, like piled up coins, the appearance. The correct answer is option C. Next question here, which was asked in the year 1991. Ferredoxin is a constituent of dash. So far we saw the photosystem 2, right? It involves photons. It's a chain of link that is it's the first link in the chain of photosynthesis. So what is ferredoxin first? First you need to know it's iron, right? Ferredoxin is an iron content. It is an iron, it's an iron sulfur protein that is the mediate electron transfer in a range of metabolic reactions. So what you can call it as, it is an iron sulfur protein. Okay, it will mediate the electron transfer in the range of metabolic reactions. What is the function of ferredoxin? The plant type ferredoxin FDS, which is abbreviated as FDS, okay, is functionally primarily helpful in the photosynthesis. They transfer electrons from photoreduced photosystem 1 to ferredoxin NADP plus reductase, in which the NADPH is produced for carbon dioxide assimilation. So it is so here if you clearly see, it is a constituent of PS1, that is photosystem 1, because they transfer electrons from the photoreduced photosystem 1 to ferredoxin NADP plus reductase, in which the NADPH is produced for carbon dioxide assimilation. Therefore, it is a constituent of photosystem 1. Okay, next question here, which was asked in the year 2017, with reference to the factors affecting the rate of photosynthesis, which one of the following statement is not correct? So they are asking us which one of the following statement is not correct here. So you're given 4 options here. Increasing atmospheric carbon dioxide concentration up to 0.05% can enhance carbon dioxide fixation rate. So here they are asking us, out of the 4 statements, which one is wrong? The first statement is correct. So what happens when there is an increase in carbon dioxide rate? The fixation will also increase. It will enhance. Look at the second option. C3 plants respond to higher temperatures with enhanced photosynthesis, while C4 plants have much lower temperature optimum. So if you remember, in the earlier parts we spoke about this. C3 plants can, their temperature is 10 to 25 degrees Celsius, while the optimum temperature for C4 plants is up to 34 to 32 degrees Celsius we saw. So here in C3 plants, the photosynthesis will decrease at higher temperature. Okay, it will not get enhanced. Always remember the temperature for C3 plants is only 10 to 25 degrees. When it happens to be a higher temperature, obviously it will not enhance the photosynthesis process. So therefore the wrong statement which is here is option B. Okay, C3 plants will not be able to enhance their photosynthesis rate when their temperature will increase. Always remember it is 10 to 25 degrees Celsius only for C3 plants. For C4 plants it's okay, you can manage up to 32 to 33 degrees Celsius. But for C3 plants, as the temperature rises, photosynthesis will not be enhanced. Look at option C and D. Tomato is a greenhouse crop which can be grown in carbon dioxide enriched atmosphere for a higher yield. Yes, it is right. D. Light saturation for carbon dioxide fixation occurs at 10% of full sunlight. That is also correct. Therefore, the correct answer to this question is option B. Okay, next question here. A plant in your garden avoids photorespiratory losses, has improved water use efficiency, shows high rates of photosynthesis at high temperatures, and has improved efficiency of nitrogen utilization. In which of the following the physiological groups would you assign this plant to? So here they are giving you a scenario where you have to read it clearly and answer it. So it's basically to do with your logic. It has nothing to do with from your book. So they're giving you a scenario, just have to match and see. See, a plant in your garden avoids photorespiratory losses. So what happens? The water use is seen to be in an efficient way. Okay, and it also shows high rates of photosynthesis at high temperatures. So this is your keyword here. Which is your keyword here? High rates of photosynthesis at high temperatures. So we saw in the previous question about C3 and C4 plants, and I clearly mentioned for C3 plants for the C3 plants I said the temperature is 10 to 25 degrees Celsius, and at higher temperatures the photosynthesis cannot be enhanced. And for C4 plants I said it is beyond 30 degrees Celsius. So it is understood photosynthesis even at high temperatures can be a good enhancement seen in C4 plants. So if you read the question clearly, you get keywords where you can make your question easily answerable. So here your keyword here in this question is high rate of photosynthesis at high temperatures. So it is obviously not C3, it is obviously not nitrogen fixer, it is obviously not CAM. Okay, it is C4 plant. Now you understand why it is C4 plant, because they can withstand high temperatures and still they can perform photosynthesis. Therefore the correct answer to this question is option B, C4 plant. So whenever you get questions like this, and if the question has to do with scenarios and to test your logic, always read it clearly so that it will be easy for you to understand and answer correctly. Next question here. Bundle sheath cells. What is bundle sheath cells? First you have to tell me that first. Bundle sheath cells are dash. So this question was asked in year 2013. What are they rich in, or what do they lack in? Bundle sheath cells are a layer of cells which are present in plant leaves. Okay, they are present in plant leaves. So where are they present? In the plant leaves. And sometimes they are also present in stems. That forms a sheath. What do you mean by sheath? Covering, which will surround the vascular bundles. In C4 plants, the bundle sheath cells contain chloroplast and they are the sites of Calvin cycle. The bundle sheath cells are the photosynthetic cells arranged into a tightly packed sheath around the vein of the leaf. It forms a protective covering of the leaf vein and consists one or more cell layers, usually which is called as parenchyma. Okay. So the other name for bundle sheath cells is that they are called as photosynthetic sites, where they are present in the leaves and the stems. When it comes to leaves, they are present in the vein of the leaves. Sometimes they even act as a protective covering over the leaf of the plant. So out of the 4 options given here, what do they lack in, what do they rich in? In C4 plants, bundle sheath cells are rich in rubesco. So I've given you the answer, right? The correct answer is option A, Rubisco. But they lack pepcase. The C2, the carbon dioxide released in the bundle sheath cells enters the C3 or the Calvin pathway, common to all plants. Thus, basic pathway that results in the formation of sugars. The Calvin pathway is common to both C3 and C4 plants. So basically, bundle sheath cells are rich in Rubisco. Okay, therefore the correct answer is option A. Next question here, which was asked in the year 2012. A process that makes important difference between C3 and C4 plants. Again, this is an easy question for you to answer, because in the previous, or in the the earlier one, we saw a scenario where you were given a passage like question where you were asked about there is a plant in your garden. It has reduced the photorespiration rate, and it has used, the water efficiency rate has increased, and apart from that, it can withstand high temperatures, and still the photosynthesis has been enhanced. So this scenario can be compared here. Why I'm telling this is because they are asking us the difference between C3 and C4 plants. You're given 4 options here: transpiration, glycolysis, photosynthesis, and photorespiration. Obviously, photosynthesis, they have not asked about the temperature here. Of course, there's a variation in temperature, but still, how that temperature, the temperature that we talk about will enhance, or it will degrade, or it will not enhance the photosynthesis process. Now, when you look at this question here, they're asking us the difference between C3 and C4 plants. The major difference is the temperature. Apart from that, photorespiration rate also plays an important role here, because in the earlier question we saw the photorespiration rate has reduced, therefore the water efficiency rate has increased, right? Photorespiration is absent in C4 plants, okay, because we saw in the earlier question, right, it has been reduced or it will be nil. So that is the major difference between C3 and C4 plants. Okay, rather it consumes energy. Further, it undoes the work of photosynthesis. There are 25% loss of fixed carbon dioxide. Therefore, photorespiration is the difference between C3 and C4 plants. Okay, the correct answer is option D. Next question here, which was asked in the year 2012. The correct sequence of cell organelles during photorespiration is. So this is actually a standard question where you cannot make any changes. Okay, so I will tell you the sequence, you just have to note it down. The sequence is chloroplast. First comes chloroplast, then comes perioxysome, and then comes mitochondria. So this is a standard question. You don't have a particular explanation for this, you just need to know the order. Okay, so the correct sequence of cell organelles during photorespiration is chloroplast, peroxisome, and mitochondria. Okay, next question here. This was asked in the year 2011. Okay, CAM helps the plants in dash. Now what do you mean by CAM? What are they plants? Because we just now saw about the plants like C3, C4 and all that, right? Now what do you mean by these CAM plants? What is the full form of CAM? I've already mentioned in the earlier part, I think, if I'm not wrong. It is crassylation, okay, crassylation, acid metabolism. Crassylation, acid metabolism is the full form of the CAM plants. They are also known as CAM photosynthesis. It is a carbon fixation pathway that is evolved in some plants as an adaptation to the arid conditions that they experience. In plants using full CAM, the stomata and the leaves remain shut during the day to reduce evapotranspiration, but open at night to collect the carbon dioxide which is present in the air. So what is CAM? I have given you a clue. Okay, the correct answer to this question is it conserves water, because just now I explained about it. When certain plants, due to certain adaptations and arid conditions, when they have CAM or when they use CAM, they close the stomata during the day and keep, so that the water is not lost because of transpiration, which is called as evaporation. Therefore, the correct answer to this question is option A, conserving water. So you call that condition as evapotranspiration. Evapotranspiration. Okay. Next question here, which was asked in the year 2011 again. In Kranz anatomy, the bundle sheath cells have dash. What do you mean by this anatomy now? Have you heard about it? Have you ever heard about it? See, when we talk about this anatomy, it is a specialized structure which describes about the C4 plants. Okay, C4 plants, where the mesophyll cells are clustered around the bundle sheath cells in a ring-like fashion. The number of chloroplasts in the bundle sheath cells is more than that of the mesophyll cells. Okay, apart from that, why is this anatomy necessary? Why should we read about this? It has been thought that a specialized leaf anatomy composed of two distinctive photosynthetic cell types is required for a C4 plant, because we all know it does not have photorespiration, therefore the photosynthesis has to be enhanced and it requires high temperatures. We provide evidence that C4 photosynthesis can function within a single photosynthetic cell that is seen in terrestrial plants. So almost I've given you a clue of what the answer is. The correct answer is thick walls, no intercellular spaces, and large number of chloroplasts. Okay, the bundle sheath cells. See here, the Crohn's anatomy specifically talks about bundle sheath cells. What are these bundle sheath cells? They are photosynthetic sites. How is their appearance? They are thick walls, they have no intercellular spaces, and they have large number of chloroplasts. Therefore the correct answer is option B. Next question here. PGA as the first carbon dioxide fixation product was discovered in the photosynthesis of dash. This is an easy question for you. You are given bryophyte, angiosperm, gymnosperms and alga. Singular form of algae is alga, so you don't have to get confused. Phosphoric glyceric acid is the full form of PGA. Okay, this was a stable product which was first identified in alga, because see, when you compare to the other plants, major discoveries or major research were carried out in algae because they resemble like plants in many aspects. They are autotrophs, they are green in color, so they have many aspects which are similar to a plant, and this aspect was first discovered, that is the photosynthesis, okay, was discovered in alga, and that is specifically chlorella. Okay, its first stable product of photosynthesis and was first discovered by Calvin, Benson and their colleagues in chlorella, which is an algae, green algae. Okay, therefore the correct answer is option D. Next question here. C4 plants, the C4 plants are more efficient in photosynthesis than C3 plants due to dash. So what are they asking us here? How come the C4 plants are more efficient in photosynthesis than C3 plants? Why? Very easy question for you to answer, because we already discussed about this a lot. The major difference between C3 and C4 plants is photorespiration. When photorespiration is reduced, what happens? The water conservation becomes high, or the use of water efficiently seems to increase. Therefore, the correct answer to this question is option D, lower rate of photorespiration. Apart from that, you also have to remember the temperature here, because C3 plants, they will not survive in high temperatures. Photosynthesis rate will not be carried out in high temperatures. Only if the temperature is low, it can be enhanced. For C4 plants, even at high temperatures, they can sustain the method of photosynthesis and they can still carry out the process. Therefore, the correct answer to this question is option D. Next question here. In leaves of C4 plants, malic acid synthesis during carbon dioxide fixation occurs in dash. Okay, so I think you will all, by all now, you have guessed the answer. It should either be mesophyll cells or bundle sheath, but I'll tell you. In leaves of C4 plants, malic acid synthesis during carbon dioxide fixation occurs in the mesophyll cells. So what is mesophyll? They are cells which perform C4 cycle, and the cells of the bundle sheath perform C3 cycle. So this you need to clarify. So you have the information here. So here if you see, they are clearly mentioned here. The mesophyll cells perform C4 cycle, while the bundle sheath cells perform C3 cycle. Remember the difference. Apart from that, carbon dioxide taken from the atmosphere is accepted by the PEP. What is it? Phosphophenol pyruvic acid, which is present in the chloroplast of the mesophyll cells of the leaves. What will it lead to? It will lead to a compound. 4C compound, which is an oxaloacetic acid. This acid is converted to another 4C acid, which is called as malic acid, and this will enter into the chloroplast of the bundle sheath and undergoes oxidative decarboxylation, which will yield pyruvic acid, which is the end product. Therefore, the correct answer to this question is option B, mesophile cells. Next question here. So it's the same question that has been repeated here, I think. Right. So the correct answer is mesophile. In the leaves of C4 plants, malic acid formation during carbon dioxide fixation is in the cells of mesophiles. Right, just now we saw. In C4 plants, C4 cycle occurs in mesophile and C3 cycle occurs in the bundle sheath of the cells. So you don't have to have any confusion. It's the same question that has been repeated. So the correct answer to this question is mesophile. So here you have a picture which depicts that. Most dicots have two types of mesophile. What is it? Palisade and spongy. Palisade is high in photosynthesis. Spongy increases the airspace for gas and water exchange. Monocot leaves will only have a single type of mesophile. The correct answer is option D. Now coming to the next question here. The C4 plants are photosynthetically more efficient than C3 plants. So continuously we are seeing a difference, and we are seeing more about C3 and C4 plants. We all know C3 and C4 plants have certain differences. That is how they survive. When I come to C3 plants, they cannot survive high temperatures. The photorespiration rate seems to be increased in case of C3 plants. It's just the opposite for a C4 plant. Now, the C4 pathway allows the photosynthesis to occur at a very low concentration of carbon dioxide, as PEP carboxylase has an extremely high affinity for carbon dioxide. So, as I told here, it is something related to carbon dioxide. So option D is not the answer. So it is something related to carbon dioxide. So it is to be between the three options here. Now they said that the pathway allows photosynthesis to occur at even the concentration is low, it doesn't matter. Still it enhances the process of photosynthesis. Okay. How is the process enhanced? If you see, it is enhanced by this enzyme called as PEP. Okay. The pathway also works well at high temperatures and light intensity, enabling the efficient photosynthesis in tropical plants. So here, if you look at the four options here, first option, the carbon dioxide composition point is more. No, because we have low concentration of carbon dioxide, the first option cannot go as an answer. Carbon dioxide generated during photorespiration is trapped and recycled through PEP carboxylase. No, see, it is not recycled because we don't get concentration of carbon dioxide in a high rate only. Whatever we get, or even if it is in low concentration, we somehow manage, the system undergoes photosynthesis. Therefore, the carbon dioxide efflux is not prevented. Okay. Even at low concentrations, photosynthesis is still carried out. So the correct answer is option C. Next question here. During photorespiration, the oxygen consuming reactions occur in dash. So this is an easy question for you to answer. It is obviously the stroma, but whether it is chloroplast or perioxysome only is the question here. Photorespiration is a process which occurs in C3 plants. Right, it occurs in C3 plants. In this process, perioxysomes, chloroplast and mitochondria will take part. So what and all are taking part in this? We have peroxisomes, mitochondria and chloroplast. Right. So now you know what the answer is, right? So during photorespiration, the oxygen consuming reactions occur in the stroma of the chloroplast and peroxisomes. Since you don't have mitochondria there, you can add also that. The oxygen consuming reaction occurs in the peroxisomes and stroma of chloroplast, while carbon dioxide releasing reaction will take place in the mitochondria. So these peroxisome and chloroplast will consume, while mitochondria will release. Therefore, the correct answer to this question is option A, stroma of chloroplast and peroxisomes. So with this, we complete the photosynthesis part 4 video. Hope to meet you in the next part of the same chapter. Thank you.